We'll go ahead and get started. Want to welcome everyone to the first. Is that an echo? Anyways, want to welcome everyone to the first, actually, of two proposal defenses today. One master's and one dissertation. I don't know what the food plans are for the second one. I'll let them figure that out. Anyways, for those who are new to our informatics graduate program proposals, basically the candidate gives an overview of his or her project proposal for either thesis, master's thesis, or PhD dissertation, and there's a presentation, some public comments, then the public is dismissed, leaving behind the committee and the candidate who may ask further questions, and then the candidate is dismissed and the committee decides whether the student is ready to move on to their thesis or dissertation. And we'll repeat this process at 2 o'clock today with Nathan. Come on in. Our master's thesis proposal candidate today is Bryan Laraway, who has a bachelor's degree in biology from Reed and has done a number of things in cellular and molecular biology, and now is seeking a Master of Science degree in the bioinformatics and computational biology track. So I will let him go from here, and we have the full committee here now.
All right, thank you very much. Okay, so today I'm going to talk about my master's thesis proposal. I'm going to be looking at a few different tools for identifying model organism models of human diseases, looking at both the models of disease and also gene candidates of human diseases. And as Bill mentioned, here's a little bit about me. I'm a researcher here at OHSU in the Ontology Development Group, which is part of the OHSU Library. I am a data wrangler for the Monarch Initiative. That's the key project I work on, where we're looking for, along similar lines, we're trying to identify models of diseases and model organisms and identifying gene candidates for undiagnosed diseases. And as mentioned, I'm obviously a graduate student here in the bioinformatics and computational biology track in DMICE. And then I also have a previous computer science training at OIT and then that biology degree from Reed College. And I wanted to start off talking a little bit about diseases in humans and just differences between common diseases and rare diseases. And an example of a few of the common diseases would be things like diabetes and Alzheimer's. You have about a prevalence of 1 out of 10 people might be diagnosed with these diseases. But then you have some more rare diseases, and these are Mendelian diseases such as cystic fibrosis, in which the prevalence is lower. And as you can see, going down the slide there, the incidence of those diseases gets smaller and smaller, to the point where in the case of ribose 5-phosphate isomerase deficiency, there was actually only one patient ever diagnosed with that disease, which would be about as rare as you can get for human diseases. And then, of course, just a little on undiagnosed diseases. These are diseases for human patients that we don't know what the cause is for the disease yet, and we don't have any gene candidates, and we're not really sure of the prevalence. It might be more, that might be just one person. It might be more than one, but we won't really know until we really dive in and find out exactly what's going on with the disease, what the underlying cause of the disease is. So the goal of my proposal, again, is to identify animal models of human diseases and then also identify gene candidates for those human diseases. And so the question I'm trying to answer here is, how do you really find, a model of a disease, a model of a human disease in a model organism? How can you determine, if the phenotypes you might see presented here, for example, obesity would be a phenotype. How exactly are you sure that is a similar expression of that phenotype in a model organism, such as a mouse or a zebrafish? And one of the key ways to go about this is to, compare those phenotypes between organisms. So if you're looking at a particular disease in humans, and the example I have here is for Alström syndrome, a few a few of the diseases, a few of the phenotypes that make up the phenotypic profile of that disease would be obesity and diabetes mellitus and insulin resistance. And if you look at similar animal models, you could see that you have similar phenotypes represented there as well, such as for the mice, you have increased food intake, hyperglycemia, and insulin resistance. And then in the zebrafish, also you'd have increased weight, increased adipose tissue volume, and then issues with a glucose homeostasis. And you can see just in between those three models, the terminology used for each of those phenotypes is a little bit different, going from one organism to the next. So the question is, when you're trying to relate these models across species, how do you determine, computationally, that those phenotypes are similar? You and I, can take a look at that, just visually, and we can tell you that yes, those all sound the same. But if you're trying to do string matching in a computer, you won't be able to retrieve those exactly without some additional help. But the key here is to consider the concept of phenotypic profiles. So if you take a genotype or a disease, you'd say that either one of those would be associated with a set of phenotypes, and the full set of phenotypes associated with that genotype or disease would be called the phenotypic profile for that genotype or the disease. And you can also do that for specifically gene variants as well. If you want to say kind of like a genotype with a single gene variant would be kind of equivalent. So the question is, if you have these two phenotypic profiles for a disease and a genotype or a gene variant, and you want to look at them across species, how do you determine that those phenotypes are related? Excuse me. And in my proposal, I'm going to look at two different methods for determining, the cross-species phenotype relations. Now, the first tool that I want to discuss is OWLSim. OWLSim provides semantic similarity metrics for phenotypes, and it takes in phenotype annotations along with ontologies being a controlled vocabulary describing a domain of knowledge that includes a hierarchy, so you can reason it up and down the hierarchy to more or less specific terms. And what you would do is, in the case of a human disease and a mouse genotype, you would take the phenotype annotations that make up the phenotypic profile for the two organisms. And you can see, on the left we have human phenotype ontology terms for the human disease, and then on the mouse side we have mammalian phenotype ontology terms. And we need to find the common term between the two. And what OWLSim will do, using the ontology, it will reason between those two, between phenotype pairs, and determine what the bridging phenotype term would be. So it would potentially have to move to be a less specific term in order to get a match. But using the ontology, it's able to reason across the differences in anatomy and physiology and species-specific terminologies to determine what that linking phenotype would be. And OWLSim would provide a set of semantic similarity scores based on just how rare the frequency that those terms might be present for the phenotype descriptions of the organism. So using this approach, if we can determine, that the set of phenotypes and the phenotypic profiles between the two species are semantically similar, we can then infer that the genotype is a representative model of the human disease, or also for the single variant gene might also be a candidate for that human disease. Now, the OWLSim approach does have some strengths and weaknesses. One of the key strengths is that it does not require a matching of orthologous genes in order to identify these similar phenotypic profiles across species. So one of the benefits there is that if you have a phenotype that is two phenotypes that are similar, but they don't involve mutation of the exact same genes, but perhaps instead mutations of genes in the same biological pathway, you could then return those phenotypes as similar, even though they aren't a direct match. So that might be a good way to explore other genes and other pathways that might influence similar phenotypes. So if you're trying to diagnose an undiagnosed disease, it can kind of broaden the scope of the genotype, the genes and pathways that you might want to investigate. One of the drawbacks, though, is that it does require sufficient phenotype annotations, and also requires sufficient integration of cross-species phenotype ontologies between organisms. And that, of course, takes curator time, and so it can be a rate-limiting step if you want to add new species to two-year comparisons. And then finally, if you're looking at genotypes or diseases that have phenotypes that manifest in a different way, especially in dissimilar organisms, say if you wanted to find a model of a human disease in yeast, since you have no anatomy to really compare with, you're going to have a much larger barrier to identifying similar phenotypic profiles. So this is where the other tool comes into play, Phenologs. The work done here, they describe Phenologs as the extension of gene orthology to orthologous phenotypes. And how this is done is you basically you examine two phenotypes in two different organisms, and you take the list of genes that are associated with the phenotype, and you search between those gene lists and identify orthologous gene matches. And if your phenotypes are enriched for orthologous gene matches, you perform an enrichment calculation using a hypergeometric probability calculation, you can then determine whether or not there's significant enrichment and determine whether or not you can consider the two phenotypes between the species to be orthologous. One of the problems with this is just if we want to compare phenotypic profiles, note that this approach is only doing single phenotype to phenotype comparisons. So in order to compare phenotypic profiles where we have multiple phenotypes, we will need to extend this algorithm to do another enrichment calculation, and this time looking for enrichment of identified phenologs between the phenotypic profiles of a human disease and a model organism genotype. So kind of similar to the previous enrichment calculation step, we'll be doing the same thing here, but instead of looking for enrichment of orthologous genes, we're looking for enrichment of orthologous phenotypes. And then in the work that the groups did towards this approach, they also developed an improved method for identifying gene candidate predictions using these orthologous phenotypes. And how they did this is what they would do is they would create a matrix, a gene to phenotype association matrix. And so anywhere on the matrix for each species, and anywhere on the matrix that you see a diamond here would be there'd be a known association between a gene and a phenotype. And what you do is you combine multiple gene phenotype association matrices and compare them between organisms. And if there's a phenolog identified between those species for that phenotype, so if phenotype 1 here for humans, mice, and zebrafish, if that's a known set of phenologs, you could then look at the genes that are associated with each phenotype in each organism and look for spots where you don't have an associated gene in one organism, but it is present in the other. So the fact that gene E is associated with phenotype 1 in zebrafish and mouse would then give a stronger prediction for that same gene possibly being associated for phenotype 1 in humans. And it might just be that for whatever reason, the state of the researchers hadn't made that association yet. So the strengths and weaknesses of this approach, one of the largest things is that we can identify non-obvious phenotype pairs. Since the approach does not require really any information specifically about the phenotype and instead works through gene orthology, we can identify phenotypes that have different manifestations. And then, of course, as I just showed you with the gene candidate, the matrix-based gene candidate approach, we can incorporate data from multiple species at once in our gene predictions for human diseases. One of the drawbacks is that it does require the identification of gene orthologs. So if a specific species you want to work with doesn't have a lot of orthologs identified across species, that could be problematic for the analysis. And then it does not account for the network effects that we might capture with the OWLSim approach, whereas this approach we're looking specifically at the mutation of orthologous genes, whereas the OWLSim approach could capture more of the network mutations that occur in the network. And then finally, one of the biggest limitations is just that gene phenotype associations. We're finding quite a few of them in different model organisms, but the experimentation that can be done in humans is obviously less. So the number of gene phenotype associations in humans trails behind that of model organisms. So with all of this, I have two different specific aims, for specific aim 1, I'm going to use these two different approaches in just mouse, humans, and zebrafish. I'm just limiting it to 3 organisms that I have good ontology integration so that I can get an equal comparison between the OWLSim approach and the Phenolog approach. And then for my second specific aim, I'm going to do an extended extended search using Phenologs, looking for candidate genes and models of human diseases. And for this one, I'm going to just grab as much gene-phenotype association data as I can get my hands on that I can clean and scrub within the time frame that I have to do my thesis work. And one of the really cool things is, for every additional species that I add, it'll just strengthen the gene candidates that I get out the other end. So just a little bit on the workflow. So for the OWLSim approach for identifying human models of disease, models of human diseases and model organisms, I would basically take the phenotype annotations for the phenotypic profiles of human diseases and model organism genotypes and combine those with the species-specific ontologies in order to relate across species and feed those into the OWLSim package, which will then return the various semantic similarity metrics for the proposed human disease and model organism genotype matches. And then likewise for the gene candidates, similar approach. The only difference here is that instead of using model organism phenotypic profiles, I'll be using the specific single gene phenotypic profiles. And then for the Phenologs approach, as mentioned before, it gets a little more complicated here because I do have to do two subsequent enrichment calculations. The first one being looking for enrichment of orthologous genes between phenotype pairs, and then the second one being looking for enrichment of phenologs between phenotypic profiles. And there I'd be feeding in the human disease phenotypic profiles and the model organism genotype phenotypic profiles to get to my model organism genotype candidates. And then for similarly for the gene candidate predictions for humans, I'll also have to do a combination step here as well, where I'll be taking my gene phenotype association data, performing the algorithm with the matrix-based gene candidate extension, which will provide gene candidates for single human phenotypes, which I will then have to combine together for gene candidates for multiple phenotypes into the phenotypic profile that would match the phenotypic profile of the human disease, and that would then provide me my full gene candidate set for that human disease's phenotypic profile. And I'm going to be doing a bunch of different evaluations, just some of the really basic ones, exploratory data analysis, looking at the total models that are retrieved by each method, looking at which models are retrieved just by one method versus the other, and then I'll be doing I'll be graphing the scores that I obtain from each method too. It'll be very interesting to see if there are models of diseases that are found to be scored highly by one method but poorly by another. I'm going to do some additional analysis to kind of take a look at those and figure out exactly what they are. For example, if a model is scored high by OWLSim only, I could use a functional interaction network, a gene interaction network, a tool known as Gene-Mania, to kind of look at the networks that are involved in that model to kind of get an idea, exactly why that one only was captured by OWLSim and not by the Phenolog approach. And then for Phenolog only, I do a manual examination of those phenotypes just to figure out, why the semantic similarity methods, we're not able to capture that model, and it might be insufficient annotation, insufficient ontology integration, or it could very well be, a very dissimilar, manifestation of phenotypes between the species. And then, of course, also do kind of like a QA test where I'll be taking asserted models of human diseases. These are models of human disease and model organisms that are kind of considered the best candidate models of human diseases, and seeing if which methods are able to successfully retrieve those models. And then for the gene candidate approach, I'd be comparing the gene candidates for the phenotypic profiles and looking at the rankings that are produced by each method, and as well as doing some additional Gene-Mania analysis just to look at the interaction networks that may be present between the say like the top 20 ranked genes, and then also for any human diseases that have known gene associations, I would then search the rankings to find out where exactly those known genes turn up in the gene candidate rankings. So, for example, for phenylketonuria, hopefully the key gene that's involved with that would be right at the top. Okay, so some of the anticipated results, I've kind of touched on that a little bit just based on what the evaluation has been doing. But I will be expecting that the two methods will have an overlapping identification of models, and that each method will also exclusively identify certain models of human diseases, and then that OWLSim will be able to capture ones that don't arise by gene orthology, whereas the Phenologs approach will be able to capture additional models that aren't semantically similar. And then the additional pathway gene candidates for OWLSim. And I'll also expect that the gene candidates retrieved by the Phenologs approach would be more highly conserved based on the requirement for gene orthology. And one of the other interesting questions that will kind of come up, and unfortunately I'm only doing 2 organisms in addition to humans, but the question would be, as the phylogenetic distance increases between the 2 organisms being compared, will the percentage contribution of one method versus the other change over time? And I would expect that for comparisons that are closer together in regards to phylogenetic distance, we'll probably have a greater contribution of OWLSim, but if we get further and further apart, we probably see a larger contribution through the Phenolog method. Hopefully that will be apparent, in comparing, human mice and zebrafish. But if I had the opportunity to, do a single-celled organism like yeast, that might be more apparent. And then, of course, the other important step, just a question of which, whether or not both methods will be able to sufficiently retrieve the validated models of human diseases, and hopefully the green section will be more covered by the OWLSim and Phenologs there. And then also, looking at the gene candidate rankings, as I mentioned before, I'll be taking a look at seeing exactly how those shake out, and then for for diseases that do have the known associated genes, hopefully they will show up in the top candidates by both approaches. And then for AIM-2, doing my extended search using the Phenolog approach, obviously with the addition of additional organisms, I'll be expecting to be obtaining additional model organism models of human diseases, and then also I'll be expecting to grab a broader and stronger gene predictions by using multiple organisms at once. And then my timeline for this work, I'm hoping to, in about a month, be able to get all the data acquisition and ontology preparation done, get everything prepared, and and start doing the actual coding. And hopefully, planning on doing about hopefully will take about a month, and then I have about 2 months scheduled, for data analysis and just seeing exactly what I got, and then after that, additional 2 months for the writings and defense. And so, in summary. What I hope to do is use these 2 different methods to identify models of human diseases and candidates for human diseases, and hopefully, and I'll be using the semantic similarity approach of the OWLSim and the extension of gene orthology for Phenologs, and I'll be comparing and contrasting those 2 approaches. And I'd like to thank my committee, who's been very supportive through all of this, and of course the Monarch Initiative, OHSU Ontology Group, and OHSU Library, and of course DMICE. Family, friends, colleagues, past and present. Thank you.
So I have a question. Let me just remind those who are new to our conference series, when you ask the question, hold down the buttons so your question gets recorded. Maybe I missed this. Is there a gold standard? I mean, what's your metric for comparing your rate of success, and is there a gold standard, and how do you plan to measure that?
Well, I mean, like the asserted models, in one sense, would be a gold standard. So being able to retrieve the known models would be a way of testing, whether or not I'm sufficiently grabbing, the models that exist. I think that's the key there. But as far as doing any other gold standard tests, I'm not entirely sure. That's kind of like one of the key ones, though.
I mean, there's a lot of metrics that are used for tests like this, like sensitivity, specificity, recall, and precision. Although you have to have a gold standard to kind of know that you're retrieving the correct model, or is this more kind of generating hypotheses for future researchers to test to see if those associations are correct or not?
Right, yeah. I mean, it is more for hypothesis generation, just kind of more of a discovery approach, and then you would follow up with subsequent, like a lab results or literature results, or, the Gene Mania functional interaction network test would be another way of examining them. Yes?
Very much. I was just thinking about, as regenerative medicine. I'm glad you have a mouse in there because it's used so often to model human diseases, but mice and rodents, they just regenerate so differently that you guys have some hard evidence. And for all we know, maybe zebrafish would be a better model. It's interesting.
Yeah, and in regards to regenerative.
Could you repeat the question? We just can't hear it on whoever's calling in.
Oh, sorry. Yeah, you have to hit the button when you ask them. But just generally, what's that? Oh, just generally, in regards to regenerative medicine, whether what might be the better models, for investigating, those regenerative medicine approaches. Would it be a mouse, or would like a zebrafish perhaps be, like a better model to study those types of injury regeneration approaches? And I'm trying to remember off the top of my head, but I want to say that, like zebrafish, might have more regenerative qualities. I mean, like one of the classic examples would be salamanders, where you can cut off an entire limb and, look at getting a full regeneration of a fully functional limb. So there might be other organisms that might be more interesting for looking into regenerative medicine, but at the same time, if we want to then translate that and interpret it to a human model, we may run into trouble unless, we can activate the same pathways that, are being activated in the salamander. I mean, unfortunately, we don't inherently have the same regenerative properties. Yes, Ted.
Nice talk, Brian. So how would like when you get your results, how do you plan to share it with the world? Do you plan to have a database of these like kind of orthologs, or?
Good question. I'm not entirely sure, like if I'll do like specific sharing of my thesis work, but the end goal, although I didn't really discuss it too much in my presentation here, the end goal would be to incorporate, so the Monarch Initiative uses the semantic similarity techniques used by OWLSim, but it doesn't currently use the Phenolog approach to identify additional models and additional gene candidates. And the end goal would be to eventually incorporate the Phenologs approach to the Monarch Initiative. And then people could then examine those models and those gene candidates at the Monarch Initiative's website using their tools. Aside from that, I mean, I'll have all of my code up in GitHub, and I don't know if I'd be creating my own repository, but I mean, most likely it would just go through Monarch.
Okay, well, if there's no further questions. Oh.
Sure. This sort of.
That was great, thanks. I wonder if we could just we went back to like about slide 22. There was a couple of things that escaped my attention when you were doing them.
Yes.
Would that be okay?
Sure. You want me to just go ahead and go over it again?
Yeah. So I mean, it's just sort of like maybe in detail, kind of each kind of like calculation that you're doing. So starting with the kind of the first gray box, and what are the inputs and outputs specifically?
Okay, yeah, sure. So starting off, you have the gene-phenotype associations from the various model organism databases. So these would just be. I did kind of gloss over in this diagram a little bit the gene orthology of it. But basically, you'd have the gene-phenotype associations. You'd have to use the original papers used. In Paranoid, but Monarch uses Panther, and we'll probably just go ahead and use Panther to basically identify the orthologs between the two species. So what's actually getting provided to the Phenolog algorithm would actually be ortholog phenotype associations, not specific gene phenotype associations, and that would allow you to compare the genes across species by the linking ortholog. So that would go into the Phenolog algorithm, where you would then do that.
Yeah. I'm sorry, that was just model organism or human as well?
I'll do human as well. I'll be looking at all of them. I mean, and then for AIM-1, of course, I'd be doing the zebrafish, mouse, and humans, and then for the AIM-2, the Wild West of whatever model organisms I can incorporate.
Okay. Mm-hmm. And then so the output of that is what sets of pairs of phenotypes. And that you then filter by p-value, is that correct? That's
correct, yes. So it would be pairs of phenotypes where there's a significant phenolog identified between the two. And kind of like before the previous step, where we're using the ortholog as a link between genes, for this one we'd be using the phenolog as links between phenotypes. And that way we'd be comparing phenotypic profiles, whereas the normal Phenolog algorithm as used is just comparing single phenotype to single phenotype. But in order to compare it to OWLSim, which compares phenotypic profiles, we need to do an extension in order to get to the same level.
So, just the first step again. So that was the first step? Is that the same as in the original McGarry paper, or did they do a kind of false discovery rate filter, or I don't recall the exact details?
Yeah. The p-values are adjusted for a false discovery rate. Otherwise, you'd have enrichment, or you'd have you'd have too many results if you didn't adjust for the false discovery rate for multiple comparisons. So yes, everything from the top down to that first blue box is the work done by McGarry in their 2010 paper, and then my proposed extension is everything down below that.
Got it. Okay, yeah, that was the part that wasn't clear when you're doing. So that's great. So your proposed extensions, you're doing the second enrichment calculation. Is that also kind of hypergeometric test?
Yes. It'd be the same test, and although I didn't discuss it here, that might be an interesting spot to look to do integration of some of the semantic similarity stuff. Just because one of the concerns I do have with this approach is that if we're doing two subsequent enrichment calculations, I'm not sure how many significant models we're going to get at the other end, since we're already doing one enrichment calculation. So I imagine the number of total models might be small, but I won't know for sure until I run the tests.
Okay. All right. Great. Thanks.
Kamal, did you have a question?
Yeah, so you'll be going through significant data sources, like going through multiple organisms. So this will involve many genes. They will appear in different models, and and obviously you'll be looking at, like in your second name, like top candidates to. But I think in some ways it might be worthwhile to keep a lot of that data and actually make it available as an annotation for something like a track on the UCSC genome browser. So what's there right now is, you can look at, for example, MGI QTLs mapped to human. There's a few things like that, but I think what you're going to build here has the potential to put like a more complete, like comparative phenotypics right onto the sort of to annotate it. First of all, do you know if such a thing exists, and how would you go about building such a thing?
I'm not sure that something exists. I know for the Phenolog work that McGarry and Woods in the two papers that they've done previously, they do have a website available, where you can calculate the Phenologs on the fly using their. You input your genes of interest in the organisms, and you can get the Phenologs that output on the other side. But I'm not sure. I'm trying to recall whether or not they had a kind of like a final data set on the other end, if that was available. I know they have the input data set available, and then they can do the calculations through the web browser. But as far as like supplying Phenologs to the other approach, it would just be a matter of running these calculations with all of the organisms that I want to work with, and then just establishing a database or identifying a existing repository that might house that. But I'm not sure if they have a existing Phenolog repository aside from the Phenolog's website.
But this would be from a more like a gene and location point of view. So you would have to come up with a way to somehow, looking from that perspective, if you wanted to annotate that particular location, like what kind of models it appears in, like which organisms, or maybe, it's going to be even maybe it can appear with your sort of this matrix comparison, it can appear in models that appear across multiple organisms, right? So
Right.
I guess you would need to think about a way to show that in a useful way.
Right, yeah. Yes.
Some of the work that they're doing is looking at ways to visualize these things on the gene, right? So
Correct, yeah. Some of the work we're doing.
Incorporate some of those ideas to that visualization scheme for annotating genotypes.
Yeah, that seems like it'd probably be the easiest approach, if nothing else, since we're already hands-on with it. That might be the best route for incorporating it.
Okay, well, we'll leave you to your committee. Thank you very much.
Thank you all.